Hello everyone, welcome back to the continuation lectures on physical anthropology. We are continuing the 1.7 part that we have done in the past class. So first we'll start with haploid and diploid cell. Okay, first I'll give you a brief what is haploid and diploid cell. Yesterday we have studied regarding cells, right? I'm sorry. Within the cells you have two types, that is prokaryotic and eukaryotic. Eukaryotic cells are more evolved cells. Okay, in prokaryotic this is found in unicellular organisms. This is found in multicellular organisms. Okay, I'm just recalling what we have done yesterday. Right, eukaryotic has got nucleus. Okay, it has got nucleus and there is no nucleus in the prokaryotic cell. Okay, after this within the eukaryotic we have seen two types, that one is plant cells and other is animal cells. What is the difference between the two? In the plant cells you have cell wall, okay, you have vacuole. Okay, and plant has also got chloroplast figment. This pigment, okay, is present inside chlorophyll, which is responsible for green color. At the same time it enables the plants to absorb sunlight and thereby generate energy. Okay, so this is the additional thing that you have. And cell wall is absent in animal cells, there is no cell wall. Okay, so this we have studied yesterday, and in that also I've told you within the animal cell there are two types of cells, that is somatic cells, other is germ cells or sex cells. Okay, somatic cells are responsible for the creation of all other body parts within the body other than gametes. Other than gametes, all other parts are resultant of multiplication of the somatic cell, or because of somatic cell division. So it is dividing and multiplying, through that all other body parts are formed other than gametes, and germ cells or sex cells are responsible for the gametes, that is sperm and ova. Sperm from male and ova or egg from female. Okay, so this differences you are clear with. Now going further deep, within the cell at the center I have told you there is nucleus. Within the nucleus you have DNA material spread like thread. Okay, so this DNA thread is known as chromatin. Okay, so this is known as chromatin. When it multiplies or when it replicates, it gives rise to two sister chromatids. This is chromatin and this gives rise to two sister chromatids. So where it attaches to one another is known as centromere. So centromere divides the chromatin into two parts. This is centromere. Okay, So when these chromatids or chromatin material, okay, so it is wound around histones like this. When it is wound around histones, okay, there are round proteins around this material is wound. Okay, so that when in condensed form it gives rise to chromosomes, which we see like this. So there are proteins, and around that these are this is what we call as chromosome. Okay, so number of chromosomes that are present in human cell, okay, number of chromosomes that are present in human cell generally is 46. Generally there are 46 chromosomes in any cell. Okay, so you have 46, right? And out of these 46, 23 comes from the mother and 23 comes from the father. 23 comes from the father. Okay, so cell has chromosomes within it, 46 chromosomes, 23 and 23. So 23 comes from the father, 23 comes from the mother. Okay, now you have seen that DNA is in double helix structure, and when you twist it, it forms a ladder, right? So this ladder only, these are the nuclear bases attached to one another, nitrogen bases in terms of A, T, G, C, etc. Right? These are arranged like this. And we have seen yesterday, messenger RNA is coming and it is taking the information. Messenger RNA is coming and it is taking the information from this DNA, and it is telling to produce certain kind of proteins and all those things. Okay, so that means there is information within DNA, and when these DNA, okay, when this DNA is wound around, when this DNA is wound around proteins, histone proteins, that means it is wound like this. Right, it is wound like this, right? And there is centromere. Now some part, that means this length, when you put it straight, when you expand it and put it, it will be something like this. Okay, when you expand this, it will become a thread again, and if you put it properly, it will become this form. And this portion, something or some part of the DNA codes for particular information okay, and this information is transmitted to the next generation. Okay, so we know that DNA is inherited, right? And this portion of where it is getting coded is known as gene. What is gene? Gene is nothing but a small part of DNA which codes for particular information, and that is inherited by the next generation. That is called gene. Now we have talked about a chromosome from mother and chromosome from father. Okay, so generally we don't have two sister chromatids. Okay, normally we have only one, but when it want to divide, that means when you want to give more, you'll multiply it. Okay, so suppose there is a cell, it is having 46 chromosomes. We'll be seeing how the division will happen, but for now understand there are 46 chromosomes. Now it want to give rise to another cell. What it will do? Now it will multiply itself, like all 46 will become 92. Okay, so these are attached, and after this again division will happen and you'll have single. Okay, so the chromosome from mother and the chromosome from father, which are having same shape and size, are known as homologous chromosomes. Okay, they have same shape and size. At the same time, within the chromosome there is a particular area. So this is the chromosome, or you can take when it is not replicated. So there is one single area which is like this. Okay, is giving certain information. Okay, suppose this information is about eye color. Okay, if this information is about eye color, so there are various colors for an eye. And okay, let's not take example for eye color because it is polygenetic trait. So what example shall we look at? So let's take this example for earlobes. Okay, so somebody is having free earlobe and somebody is having attached earlobe. Suppose this gene is giving information regarding whether you have to have free earlobe or whether you have to have attached earlobe. So for some people you have attached earlobes. Okay, this is earlobe, and for some people it is attached. Okay, dominant is free. Okay, so you see in most of the people have free earlobe. That is dominant because it's a dominant trait, and we'll see what is dominant and what is recessive. Let's not get there now. Okay, so there is a gene and it is giving certain information. Okay, in other chromosome which is coming from the mother, so suppose this is the gene part, it is telling for what kind of earlobe you should have. So mother is telling a particular thing, and in the same position you will have a gene. Here also the gene talks about what kind of earlobe you should have. Okay, so shapes are similar, and even the position of genes or the position of information for particular trait is same. Okay, for the position, give this number 51, suppose random number. So this is the 51st position which talks about earlobe. In father also you will have 51st position which is talking about earlobe. And most important thing is that if you go to 51 position, it talks about earlobe, but the position, even though it is same, but information is not same. Okay, earlobe, here it might be free, here it might be attached, or both can be attached or both can be free, anything can be there. Okay, information need not be same. Okay, information need not be same, information can be different. Okay, now you have understood, right? One comes from the father, one comes from the mother, and there you'll be seeing the positions of a particular trait. Okay, position of genes for a particular trait will remain same in the both, but what information that gene is carrying will vary. Okay, since there are two different genes, so variations of genes which will tell the trait for a particular thing. Okay, so there are two genes, two variations of genes, okay, two variations of genes for a particular trait are called alleles. So alleles, which we discuss generally, are nothing but variations for a particular trait. Okay, variations of gene for a particular trait are known as alleles. So never get confused between genes and alleles. Okay, alleles are also genes, but when you are talking about a particular trait, if you are talking about eye color, for eye color, one gene, you have two chromosomes. One gene comes from mother, one gene comes from father, and when you are talking about them together, okay, we call it as alleles. Okay, so when you're talking about them together, you call it as alleles. Okay, so I think now you are clear with what is chromosome, how many chromosomes we have within the chromosomes, where is the information coded, which is known as gene, and gene has got variations because you get one from mother, one from father, that is known as alleles. Okay, so DNA, gene and allele. So some part of DNA is known as gene. Okay, don't get confused. I have written DNA. DNA includes this entire length. So this is DNA. This complete is DNA, and within DNA some portion codes for particular information, that is gene. Okay, so this while explanation I have written it. Okay, so some portion which is coding for a particular information is known as gene, and you have variations with respect to information from mother and father, that is known as alleles. Okay, just watch this video of haploid and diploid cells, right? So I have told you somatic and germ cells. Now we have seen our human body consists of 46 chromosomes in all the cells, but in sperms and ova, which are produced as gametes, these will be having only 23 chromosomes in them. Okay, in their cell, if you see the sperm cell or ova, you will have only 23 cells. Why? Because sperm and ova will fuse with each other, then 23 will combine and will give rise to 46. In order to maintain constant number of chromosomes, okay, in every generation, so the kind of division of cell which happens in germ cells is different. Okay, so the division here is known as meiosis, and either here we call it mitosis. Okay, so what is meiosis and what is mitosis we'll be seeing in a greater detail, but you need to understand gametes have only one set of chromosomes, and whereas what do you call somatic cells have two sets of chromosomes. Okay, so that is 46. Now you might be thinking that, so here 23 chromosomes are only there, whether father's chromosomes will go or mother's chromosomes will participate, because it might be little confusing. You know, male and female, they combine to give rise to children. Okay, so there is a male child and there is a female child. Okay, so now male child has 46 chromosomes. Okay, 46 chromosomes consisting of 23 from mother, 23 from mother and 23 from father. Right, now in this male child sperm cells, which of them are going to come? So this would be the doubt for most of the students. This we will understand while doing mitosis and meiosis. There you will see a process known as chiasmata or recombination. Chiasmata or recombination. So this process where these two will interact and give rise to 23 chromosomes, which are combination of these two. Okay, so which are combination of these two, so which are combination of mother and father. So those two will combine and will give rise to new one. Okay, so that process you will understand better, but there will be generally there will be a doubt. So now father has 46, 23 from mother, 23 from father. So while giving to the child, whether he'll give his father's or mother's, so that there will be generally a doubt of it. So it will be fusion of both. So now just go through this video of haploid and diploid cells, and after this we'll be seeing mitosis and meiosis. Okay, haploid and diploid cells are nothing but haploid cells are having only one set of chromosome, and diploid cells are those that are having two set of chromosomes. So two set of chromosomes is seen among somatic cells, and one set of chromosome is seen among gametes. Okay, so just for your visual understanding, I have added a video. Just go through this.
In this video, we'll discuss haploid versus diploid cells.
So you have understood what is haploid cell and what is diploid cell, right? So they are different in terms of the number of chromosome sets that are present. There are only 23 in the haploid cell. Okay, in the diploid cell you have 46. At the same time, there are no homologous chromosomes. Homologous chromosomes in the sense one that is from the mother, one that is from the father, which are coding from the for the same information. Okay, so they have genes which are coding from the same information known as alleles. So that thing is missing, right? Homologous chromosomes are not present in the, what do you say, that haploids. At the same time, what is the role of haploids? Haploids helps in the promotion of or production of germ cells, whereas diploids helps in the production of somatic cells. Okay, so all the somatic body parts are produced through the multiplication of diploids, and whereas all the germ cells are produced through the multiplication of haploids. Okay, how the cell division happens? Within the human body, we are going to learn now. At the same time, see, if you are becoming this big from the small size of an embryo, you can understand that some kind of multiplication could have happened, right? Now there are cells of two types we have seen. The somatic cells will multiply, these cells will multiply, and there are germ cells or sex cells. You can call these are sex cells. These will also multiply. Okay, so what process they are going to adopt is in this process of multiplication. Okay, so for somatic cells, they are going to adopt a process known as mitosis, and these cells are going to adopt a process known as meiosis. Okay, so this is with respect to the types, and within these what is going to happen. Okay, so before a cell has to multiply, okay, so when you are giving birth to a new one, you should have enough amount of energy. Okay, so suppose if you are multiplying, then you have to feed the next one also. So there is a need for enough amount of energy that has to be present, enough amount of nutrition that has to be present. Okay, so first cell ensures before division, okay, before division cell ensures that it has enough amount of nutrition. That is the reason, if you see, when you don't provide proper nutrition, right, the children doesn't show proper growth, right? There'll be generally, you see that in case of marasmus or kwashiorkor, so there is thinning of people, right? What is the reason? That enough nutrition is not there. That is why there is no multiplication of the cells and they are thin. Okay, so enough nutrition has to be present. Okay, and at the same time, the first phase, okay, first it ensures that all the things are right and I have good nutrition. Now I can multiply. So first phase, it prepares itself for division. That is known as interphase. First phase where cell prepares itself for division is known as interphase. In this, what it will do, we'll see in the video so that you can better understand. Okay, so within the mitosis there are 4 phases of division: prophase, okay, metaphase, anaphase, telophase. Okay, so in the meiosis also the same phases are present and they will repeat twice, and how and what we'll be seeing in this video. Once you go through this video, then I'll explain each and every step of it. So this is the comparison of mitosis and meiosis at a time so that things are clear. Okay, so this you can see. So now after seeing this video, you will be better able to understand what we are going to learn. Right, first is mitosis and the second is meiosis. Right, in the process of mitosis, right, in the mitosis, what happens first, like before cell going for division, it ensures that it has enough nutrition. Okay, at the same time I told you the chromosomes are not in pairs. Okay, they are not in pairs of sister chromatids, they were in single chromatin. Okay, they are in terms of single chromatids and not sister, they doesn't have a pair of sister chromatids together. Okay, so now what happens, like this you have 46. Okay, you have 46, 1 2 3 until 46 you have chromatids. Okay, and now 23 out of this are from mother and 23 are from father. Okay, now since they are going to divide, next, that means they should double themselves. Okay, so that's why they will create a same pair. Now this will multiply a plus a, that will gives rise to. Okay, so these two are same, this is having the same information as this. Okay, but in germ cells you'll see that mother is having different information, father is having different information. So like this, now they'll multiply and you'll have 46 as such. Okay, 46 as such, and you'll have 92 chromatids. Now you have 46, when it is multiplying into 2, okay, that is 92 chromatids, but we still call them as 46 chromosomes because they are having the same information. Okay, so these two are having same information, right? So this is the first thing. Now it is getting ready to prepare, that's why it has multiplied itself. Then after that you have 4 phases: prophase, metaphase, anaphase, telophase. Okay, in the prophase, that means first phase of division, so proto division, before the division happens. In this phase what happens, the chromosomes which are in terms of chromatid will start winding itself and it will try to condense. Okay, so it will try to condense and will start to appear. Chromosomes will start to condense, and now you can see them. When they were thick fibers like structures, you can't see them, but during this phase, okay, slowly they'll start appearing. Okay, at this point of time what happens, there are something present outside the cell that are known as centrosomes. Okay, centrosomes, right? Centrosomes are present in the outer part of the cell. Now centrosomes will come in. Okay, centrosomes will come in, and these centrosomes are having spindles, that means tubule like structures. These are spindles, okay, these spindles will attach to the microsomes. Oh sorry, not microsomes, chromosomes. These spindles will attach to the chromosomes. Okay, if you see this, it is more visible in the video. Okay, so these will attach to the chromosomes like this, and they'll start pulling it away. In the second phase, what happens in the second phase? Chromosomes, metaphase means middle, okay, chromosomes will come to the center. First chromosomes are random, in the second phase chromosomes will come to the middle. Okay, chromosomes will come to the center, and this centrosome is having spindles, and these are attached to the centromere. Okay, so these spindles are attached to the centromere. In the anaphase, what it will do? Ana means, remember that, away. Okay, so it will take away. So this will be spread and will be taken away, and after that in the fourth phase, telophase, they will reach. Okay, in the fourth phase they'll reach the ends, and after that the spindles will disappear, and after that you have cytokinesis, which divides the cytoplasm. Okay, cytokinesis. So in the cytokinesis, what happens? It will divide the fluid that is present into two parts, and through that cell division will complete. Okay, so this is with respect to mitosis, right? Mitosis has 4 phases. So in the meiosis, what happens? We'll see. Now so you can see here, first there are diploid cells. Okay, so in meiosis also, in the beginning you will have diploid cells, but the end, when they come up, it will be haploid. So now you have 46 chromosomes. These are getting multiplied. So you can see. Though there are 92 chromatids, there are only 46 chromosomes, right? And now if you see further, what happens in the interface? There is duplication of the DNA. So DNA is multiplying itself. Okay. So just since it's not possible to draw all the chromosomes, they are representing only few to show, right? So you can see in the prophase what is happening, like before the cell division happens. So what is happening now? The chromosomes are trying to appear. Okay, they are condensing. Okay. So in meiosis, what happens is, now I've told you homologous chromosomes, right? Homologous chromosomes are the same kind of chromosomes that are present both in mother and father. Okay, so there will be same size, same shape. Now there are 23, right, which we were seeing, one from mother, one from father. Okay, so they are multiplying. Now you have 92, right? You have total 92 chromatids and 46 chromosomes. Now out of 46, now 23 pairs are formed. Okay, so now 23 pairs are formed here. Okay, see here 46 will arrange line by line. Now you start from the beginning, 1 2 3 4 5 6 till 46 it goes. 46 chromosomes are arranged in a line over here in the metaphase. Okay, they'll come to the middle, and during this 46 will happen. So the things that you are seeing here is, so these are 46. 1 to 46, they'll arrange themselves. So one is from mother, one is from father. Okay, so from mother you will take one, from father you will take one, and this are arranged. So in this prophase, what happens in meiosis? In meiosis, what happens is that homologous chromosomes from father and the mother, they'll come together and they'll start as pair. Now, instead of having one chromosomes at the center attaching to the spindles generated by the centrosome, okay, this is the centrosome. Instead of attaching one, okay, here what happens, one thread will come from here, one thread will come from here, they will attach to one chromosomes at one centromere. Okay, so let me show you. So there is a chromosome. Okay, sorry. Okay, so now there is centrosome. This is centromere. I have told you this is centromere. So these centrosome, this part is known as centrosome, which you are seeing in pink color in the diagram. Centrosome has got spindles. Now this spindle will get attached here and this spindle will get attached here. So here in the prophase of mitosis, centrosome spindles are attaching to the same centromere from the both sides. Now what happens in the meiosis is there will be pair. Okay, so now they are as pair. Now like this you will have different pairs. So there are centrosomes. Now it will attach to this centromere, it will attach to this centromere. Okay, so this will attach over here. So here from 1, 2, 3 till 46 you have. Till 46 you have. And now what happens? 46 are dividing into 2. Okay, so for one will go to that side, one will go this side, and you will have 46 chromosomes. Okay, so 92 chromatids were there. One part is enough, right, to get the information. One chromosome part is enough. So this was what did it do before dividing? It multiplied itself and it made into 2. Now it is again becoming 1. Okay, so 46 will go this way and 46 will go that way, and after division again the cell will have 46, 46. So that means 46 chromosomes are present in the next cell as well after the cell division. Okay, but here what is happening? You are having pairs. That means 1, 2, 3, 4, like this you have all those things. That means only 23 are there. Okay, 23 pairs are there. Now what happens? Each one will go to one end. That means one chromosome is moving towards one end. Okay, so these are two different chromosomes. Here it is same chromosome. Okay, so here it is same chromosome because you have 46. 23 are down from mother, 23 from father. But here you have one from mother, one from father. Okay, so here each mother and father is getting separated, but here one mother and one father is getting separated. But before appearing like this, there is a process known as recombination. Okay, there is a process known as recombination. Now they have come here, right? So first consider that this is from the mother and this is from the father. Okay, suppose there is yellow color over here. and there is yellow over here. So that means there is some information over here which is present within the father, and there is information within the mother. Now what will happen, these two will overlap like this and they'll exchange the things. Okay, what is getting exchanged or they're recombining, we don't know. Okay, now this will move over here. Now you take, so these are the new ones afterwards. Okay, this yellow part moved from father to mother. Okay, now it is moved, it has moved from father to mother, and from mother something, suppose there is something over here, now that will come to father. Okay, so this process of exchange of information from mother and father, okay, will gives rise to two new chromosomes, okay, consisting of different information. This will happen before coming to the center. Okay, so that process is known as chiasmata, or okay, crossing over or recombination. They are using different terms, but it is nothing but before coming to this center, these chromosomes which have come to the center are the new ones after recombination. After recombination, whatever the new ones that have come, okay, so whatever the new chromosomes that are formed, that will come to the middle. So now you have to understand one thing. We had a doubt, right? So now when chromosomes are going to the child, whether it will be fathers or whether it will be mothers, okay, it is combination of mother and father. Okay, right, two chromosomes are coming and they'll attach like this, and after that they'll exchange whatever the information that is possible. Okay, it is natural, it happens naturally, and it is not known how the exchange happens. So since it happens naturally, okay, so this exchange happens, that process is known as recombination, and that gives rise to new types of chromosomes from what has already been there, and these chromosomes are divided into two. Now this will go at one end and this will go at one end, and the same process that happens in the mitosis is repeated in the meiosis also. Okay, so now here they'll separate and they'll form 46 based chromosome cell, and now you have 23 chromosome cells. Okay, so now 23 are formed, but you have 46 chromatids, right? Now what happens again, just like mitosis that has happened, okay, where 46 gives rise to 46, here now again 23 will give rise to 23. So 46 was there initially, and 46 gives rise to two cell, 46 and 46, and here it will give rise to 23 and 23 in the next division. Okay, now watch the video, you will understand better. So now one from mother, one from father will come and will form homologous pair, and now what happens, they'll sit over one another and they'll exchange. Okay, now exchange of information will happen, but it will be with respect to same location, right? Suppose homologous, why do we call them homologous? Because they have same structure, same shape, and this information for a particular trait is present at the same location, right? So if there is 23 over here or 51 or whatever, you can take whatever the location, same location will have information for same trait. Now only that position will exchange, it's not random exchanging. So this recombination will happen, but recombination will happen in such a manner that location remains the same but information is exchanged. Okay, that is known as recombination or crossing over. So now you can see whatever the information that was there from mother has been moved to father, and from father to mother. Okay, so where these are getting exchanged we don't know, but this exchange will happen. Now the chromosomes that have come from mother and father in the initial is not same as the chromosomes that are going. It is combination of both mother and father. So now what is happening during the metaphase? They'll come to the middle, and during the anaphase they'll move away, and during the telophase they'll come to the end, and cytokinesis will happen and it will break it into two. Okay, so now so now in the anaphase what is happening here? Only one chromatid is moving towards the others, but here entire chromosome or pair of chromatids are moving towards the other, and because of that you'll have 23 chromosomes only in the coming cell. So here only chromatid is being pulled, here entire chromosomes is being pulled. Okay, chromatid is different from chromosome. Okay, in the cytokinesis it will divide the cytoplasm, because before itself there is a formation of membrane, and after that division will happen. Now you have seen that mitosis and meiosis in the first phase, but mitosis, here the cell division is complete and it gives rise to new cell which it wants to be 43 chromatids. Okay, so which has 46 chromatids, not 43, right? But in case of meiosis there is a second phase which is same as mitosis. Okay, so now this will come to the middle, these chromosomes will come to the center, these chromosomes will come to the center in the metaphase, and they'll divide in anaphase, they'll move away. Okay, again you will get 23. Okay, so 20. So now you have identical cells which are diploid and which are having two sets of chromosomes. So again here in the prophase cells will condense, and after that you need to understand here there is no homologous pairing or crossing over in the second. It is just like mitosis. They'll come, they'll divide and they'll go, and you will get 23 chromosome cell which is known as haploid. So now one basic difference between the mitosis and meiosis is that when the cell multiplies it gives rise to two cells, but here when the cell is dividing, or when cell gets divided, it gives rise to four cells. Right, so these are known as ova or egg, right? So these are not identical, four are different because different chromatids are going to different place. Now you see that a sperm cell, when it comes and attacks or the combines or fuses with the egg cell, when the sperm cell fuses with the egg cell, what kind of child is going to be born is depending upon which egg it is going to fuse with. right? So that's why there can be variation with respect to the child that is going to be born. Okay, so these are non-identical and these are gametes. Okay, so male, from male you can see sperm will be coming, and when these are in case of female you can take it as egg. Okay, so these can be acting as sperms carrying different chromosomes, and these can be acting as eggs where the sperms can come and attach to it or fuse it. So these are both, it can be gametes, sorry, both it can be ova as well as sperms. These things you need to keep in mind. Okay, so this is with respect to mitosis and meiosis. Okay, so I think you are clear with mitosis and meiosis. With this we are done with cell division. Okay, so one more important thing I thought of telling you people, which is known as thrifty gene. Okay, so there is something known as thrifty gene. I'm sorry, extremely sorry. So so he's the one, is a scientist from America. He took interest in studying the reason for increasing the type 2 diabetes. Okay, type 2 diabetes. So what is type 2 diabetes? We'll get to know about it in greater detail, but there are different types of diabetes. One, you can understand that one version is type 2, which is generally seen due to the lifestyle. Okay, different lifestyles that is present. Okay, so when it is increasing, this hypothesis of thrifty gene has been put forth by James Neal. What he has found is that, see, within the human body there is a gene known as thrifty gene. Okay, this gene is responsible for allowing to store extra fat, within the cell. Okay, it enables the cell to store extra fat. Okay, at the same time it was an advantage as a part of evolution. Okay, it was an advantage as a part of evolution when it has come. So this gene was there, it was letting the cell to add more and more fat. So when it can add more and more fat, what happens? This will help you have stored energy during the famine, right? So since the amount of food that used to be available, the resources that used to be available was not constant, and there is no such mechanism to store in those times. In those times, what used to happen, this kind of mechanism developed by the thrifty gene was advantageous. But now what has happened now, that issue was not there. The issue of famines was not is not present with the proper development of the states and the other organizations of human society. So through that, what has happened, there was no necessary for storing excess fat within the body because there was no such thing of famines. Okay, so now what has happened, the same gene which was advantageous previously, now it has become a liability, because of which storing of excess fat, okay, and improper lifestyle is leading to the type 2 diabetes. So this theory or this hypothesis of thrifty gene was developed by Neal, and this is with regarding thrifty gene, and this question has come in exam already. Okay, so they might ask something new, so that's why you need to follow the updates anything that has happened. So like, okay, so Plabo's recent theory on evolution, and he has found out that certain mechanisms which can help in understanding evolution. So that thing you need to go through. Recently he has received the Nobel Prize for that, so it may become important for UPSC as well. So they may not ask what he has done, but they may ask something related to the evolution. Okay, so that part anyhow we'll be covering in the other topics. I'll be taking up them, but what you need to understand is with respect to cell, mitosis, meiosis, and we have seen haploid, diploid cell, and we have also seen that protein synthesis, like how from mRNA goes and it gives to the ribosome, then from there how the proteins are synthesized. That is protein synthesis. And there is one more thing that I have to tell you. So that is known as central dogma theory, central dogma theory of the cell. So it is nothing but central dogma theory says that it is one way functioning of the cell. What happens? Okay, so within the cell, DNA leads to the formation of or multiplication, replication of DNA happens, and from DNA, when it divides, it gives rise to RNA. So from DNA, RNA forms. Okay, so from RNA, which takes information and gives rise to protein, right? So the direction of forming remains the same throughout. That's why within the cell this theory is known as central dogma theory, which talks about the synthesis of protein from the DNA. Okay, so replication of DNA, after that transcription, and then translation. Okay, so this involves a particular direction of formation, and this concept is known as central dogma of cell theory. Okay, so this is one more thing that you need to know. You need not remember all these things. Whatever we have done throughout this, you need not remember, because these things are not asked directly in the exam. But now when we'll be studying unit 12, unit 11, or any other unit as a part of genetics, there we'll be using the words recombination, crossing over, okay, homologous pairs, alleles, genes. Then to understand what we are talking exactly, you need to have idea of this. Okay, so for that reason I have took some time in order to explain all these things, or else directly using the board also you can teach these concepts, but when you see them you'll remember for a long time and you won't get confused what exactly is happening. Okay, this is all with respect to 1.7, and in the next class we'll be starting with 1.4. okay, that's all for today. Thank you.